friends, welcome back to another video session from Shomu's Biology, and we've been talking about the immunology and the different parts of immune system and how immunology is constructed with all the immune system cells. In the last video we talked about the process of antigen presentation and the role of antigen presenting cells in contacting both the cell mediated immunity as well as the humoral mode of immunity. Now in this video I want to talk about MHC1 and MHC2, because with the help of these two different surface receptors, antigen presenting cells can present antigen. Okay, so what are those things? MHC1 and MHC2, these are nothing but kind of a glycoproteins that are found on the surface of eukaryotic cells with nucleus. Okay, now there are two types, MHC1 and MHC2. All the nucleated cell in our body contains MHC1. They have MHC1 on the surface because we have the cells are filled with all the glycoproteins receptor on the surface. But this MHC1 is different for different person. It is tissue specific. For different tissues, this MHC structure and its different length of the chain and the amino acids that are present in the chain to make the MHCs will vary. So that is why the name is different, like major histocompatibility complex. Why this name? Because that when we discover this glycoproteins, it's found out during the tissue grafting from one person to the next person, or organ transplantation from the donor to the recipient. Now when we're transferring an organ from a donor to the recipient, all the MHCs should match, otherwise the recipient cannot accept the donor organ, because it's about again foreign and what is self, because it's distinguished by our immunity, whether it is a single self molecule or it's a foreign molecule. Because it's coming from the outside, so it's a foreign, they recognize that as a foreign particle. The only way they will not recognize it as a foreign particle if it carries the same sort of receptors on the surface, because all this immune system that we are looking at, it's a beautiful example of cell signaling going on at a remarkable scale, because all those small molecules, all those cells and everything that are there, they are interacting with the help of different cell surface receptors. They are interacting by secreting chemokines, which are the signaling molecules, the chemical factors. So what happens actually, this receptors are playing very vital role. That's why the name comes major histocompatibility complex. So whether these glycoproteins are distinguished and they determine whether a tissue graft will be rejected or it will be accepted. That's why the name came. But now if we go back to the discussion what we are talking about, all the nucleated cell carries this major histocompatibility complex 1, but they are lacking major histocompatibility complex 2, because MHC class 2 is only found in antigen presenting cells or APCs. And what are the antigen presenting cells? In immune system we know macrophage, dendritic cells and B cells, they are known as antigen presenting cells. Why? Because they have a specific function which is very special, that if there is a pathogen, they will engulf that pathogen with phagocytosis. Then they will break that pathogen down into small fragments which are known as antigens, and they will showcase that antigen by loading them with a receptor and show them to rest of the immune system cells. But that receptor where they load this antigen are MHC1 and MHC2. That is the job, right? But all the cells have MHC1, so if required all the cells can supply and load the antigen fragments to MHC1 and showcase, but only macrophage, dendritic cell and B cells can have MHC2. So when they start to fragment the foreign invader or pathogen and showcase that to rest of the cells, then they will need MHC2, and they only have that. Rest of the cells don't have MHC2. Okay, so now let's talk about this whole process in much more details. The difference between MHC1, in the last video, if you are looking this video and watching this video sequentially, in last video we talked about the antigen presentation, and we know requirement of both MHC1 and MHC2 are important, because MHC2 will help to activate some more T cells, some more T helper cells that will help in bringing more macrophages, more dendritic cells. So more antigen presenting cells will be bought, as well as this interaction between the antigen presenting cell with the T helper cell with the help of MHC class II molecule can also help in activating the B cells and start producing antibodies. So all these things are brought with the help of MHC2. But what is the job of MHC1? The function of MHC1 is to designate a specific cell of our body as sick, as weak, and it will be killed. So there are two options when there is something wrong going on in our body. First thing is that foreign pathogen enters, pathogens start to spread. Now it's not killing a cell or it's not damaging the cell then and there, but it's spreading. So what we want to stop the spread of that pathogen. So we'll engulf that pathogen, break them down, present them with MHC class II to the rest of the cells. T cell will be activated, it will further activate rest of the antigen presenting cells, and it will activate the B cells. But on the other hand, if some of our normal body cell is diseased, it's infected, and there is no way that disease can be fixed. For example, if it's invaded by viruses and the cell is filled with virus particles, which is drawn here with these red dots, let's say those are virus particles, they completely invade the cell and infect the cell. There is no way the cell is going to survive. In those conditions, to limit the damage, sometimes we need to kill our own body cells to prevent that damage there, because if we kill that cell, this disease cannot be spread to the nearby tissue. So to do that, in that case, those targeted cell or those sick cell, they have MHC1, because all nucleated cell have that MHC1. So they will showcase some part of this component outside, those fragment of antigens outside, or they will sometimes showcase the alteration of lipids in their membrane, which give the signal to the nearby cell, which could be natural killer cell or cytotoxic T cell, to guess that this cell is infected. We need to take an action. For example, here if MHC1 loads some part of the fragment of pathogen and showcase outside, so this cell is now very sick, it's showing a flag outside that I am sick, please kill me, to prevent the damage to the rest of the tissue. So cytotoxic killer cell will come, it will interact with the help of T cell receptor with MHC1 loaded with the pathogenic portion. It starts secreting some chemical factors known as perforins and granzymes that are going to ultimately kill the target body cell. These are the job of both MHC1 and MHC2. Now another thing I must tell between the difference between MHC1 and 2 is that MHC2 is provided by this dendritic cells or macrophage, all these antigen presenting cells. They will interact with CD4 type of T cells, while MHC1 interact with CD8 type of T cells. CD4 type of T cells are known as T helper cells, because the job of this T helper cell is to activate some more T helper cells, is to activate B cells, is to proliferate macrophages or any other antigen presenting cells. While this job of CD8 plus T cell is known as cytotoxic T cell. So let me write that, cytotoxic T cell. So it's toxic for cells, right? So these CD8 T cells are cytotoxic T cells. So the job of this cytotoxic T cell is to kill the target cell. Okay, that is another functional difference between MHC1 and MHC2 and how they engage in interaction. Now if we go in more details about the structural and functional process of and processing of MHCs, there are some differences present between them. So for understanding, I must erase this portion, and one thing I can tell you is that this MHC1, it has a structure of, let me draw it in a very basic drawing of the structure. It has two different chain. One is the alpha chain, which is the proper full alpha chain, and a partial beta chain, a partial beta chain. That is the structure of MHC1. But if you look at the structure of MHC class 2, here we have a proper alpha chain along with a proper beta chain. Okay, this is the structure. This is the structure of so proper alpha chain and beta chain together, and here is only alpha chain and a partial beta chain for MHC1. Now how they prepared these structures? Now the thing is, inside the cell, so let me draw this whole idea. I'm not going to talk very details, but I'm just going to give an overview of this process. If this is the cell, all this processing of antigens and loading the antigens on this MHC taken place in the endoplasmic reticulum. So I'm drawing the endoplasmic reticulum here. Let's say these are the regions of endoplasmic reticulum. And normally what happens in the cytosol, there are proteasome. Proteasome are the giant machines that are present in the cytosol of eukaryotic cell, which can break down proteins into smaller fragments. So what happens actually, let's say there is an engulfment of a pathogen. So green is the color of the pathogen, a bacteria for example. They engulf this bacteria inside. After this engulfment, it is released. So the fragment of, so normally what happens after this engulfment, this vesicle will be fused with lysosome, and you know lysosome is filled with all those proteolytic enzymes that are going to degrade some portion of this fragment of the pathogen, and it will be released outside. For example, if it's a bacteria, it's a, let's say, gram negative bacteria, so they have a LPS, lipopolysaccharide layer outside. After this breaking down of the components, some part of these LPS layers are fragmented outside. So even though they fragment these portions, it could be LPS, it could be any protein portion of the bacteria or any other part of it. So whatever it's, let's say it's a long chain of a peptide. Now that long chain will be further broken down by the proteasome into very tiny fragments. That is enough to be loaded onto the MHC. That is the process. They engulf the bacteria. So let me write it as a bacteria. They engulf the bacteria, break them down, fragmentize it further with proteasome, and then it's ready. Then what happens? On the other hand, in the endoplasmic reticulum lumen, inside the lumen of endoplasmic reticulum, they have MHC complexes prepared. Let's say this is MHC complex 2. They make this complex ready. How exactly they make the complex ready? You know, in case of MHC2 preparation, they have both alpha and beta chains prepared, and they have this portion of binding of the antigen. That portion is not free. However, there is a specific accessory protein that blocks that binding region from the beginning in this endoplasmic reticulum. It blocks this binding region normally. Now once they fragmentized all those bacterial fragment and it's taken inside to the reticulum lumen, it's taken inside the reticulum lumen with the help of different transporters that are present here. The transporter present between the reticulum membrane and the cytosol. When it's open, they can take all those fragments of bacterial antigen inside the endoplasmic reticulum lumen. Now once they bring them, then this invariable peptide chain that we have, it is broken by subsequent release of proteolytic enzymes. But still, even after it's broken into fragments, it still have some portions left, and this portion is known as clip. Okay, the portion of the invariant chain peptide still present in the attachment or antigen binding side of MHC2. Now what happens actually, once this fragment is ready, the clip is present, the clip is present, and this clip containing MHC class 2, this clip containing MHC class 2 is then released out. It's then, let me show the bulging, it's clipped out this is the portion of the clip. So it's like bulged out. Once it's bulging out, then some part of this bacterial antigenic fragment replaces the clip and properly place themselves in. Remember the portion. So normally at the beginning this is filled with some invariable peptide. Then once it starts releasing and proteolytic enzymes are secreting, it's getting clipped. Some portion is still attached to the antigen binding portion. As now they are coming out, and also the fragments of bacterial antigen start, the concentration of fragment of bacterial antigen increases, it will dissociate this clip and the antigen will occupy this place. Okay. Now once the antigen occupies the place, that fragment MHC2 loaded with bacterial antigen is now will be fused with the membrane, and as a result of that, what we will see is antigen attached on the surface of this antigen presenting cell with the help of MHC class 2, this black one. This is how they represent the MHC class, the antigen with the help of MHC class 2. The process is kind of very similar with MHC class 1, while they, in that case they do not have any invariable peptide or binding with the antigen binding site. That site is free from the beginning, and once they make the fragment of antigen ready, they simply load that on the top of MHC1 and then they provide it to the surface of the cell. So this is how the MHC processing is done. Okay. Remember, this processing is very important, because if they do the processing with MHC class 2, then they are going to interact with CD4 type of T cell, which are T helper cells, and the consequence we saw earlier. But when they are interacting and load them into MHC class 1, they are going to release it outside, and in that case the cell will be killed with the help of cytotoxic T cell, because MHC1 will interact with CD8 type of T cell, which are cytotoxic in nature. So that in a sense is the process of antigen presentation and the role of antigen presentation in major scale between both MHC1 in case of MHC1 as well as in case of MHC2. But one another thing I should tell is about these antigens that will be presented. Now there are different, like you can tell me that there are multiple fragments, there could be cellular fragments even that are present inside the cell sometimes. The cellular protein is also being degraded by the proteasome. So why not the cellular protein is loaded onto the MHC2 by chance or by mistake? Yes, good question, but the problem in this case is that it does not happen all the time. It's, in fact, most of the time this problem don't have any issue. The reason for that is the binding site for MHC2 is very specific, remember, because the MHC2 antigen binding site is occupied with the clip peptide until and unless they find proper pathogenic antigen to be attached. Only if a pathogenic antigen is found, then only the clip peptide will be released and the antigen will be placed. Now the question is, how come a cell recognizes a pathogenic antigen? Now the answer for that, they don't have to recognize a pathogenic antigen, but they can recognize their self body fragments. So they don't load the self body fragment in MHC complexes. They avoid that, and rest of whatever thing they don't know about must be from the pathogenic components. Another thing is, in the MHC2 and the binding groove of the MHC2, there should be a specific length of amino acids that can be accommodated. More than that will not be able to be bound, less than that will not be able to be bound. So the only specific length of amino acid long peptide can only bound, and that controls what is going to be interact and what is going to be attached with that MHC2. That's the idea. The complex of MHC1 is different, but MHC2 is more important in the perspective of immune responses that we know. Okay, so that in a sense is the difference between major histocompatibility complex class 1 and class 2, also known as MHC1 and MHC2. So if you like this video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more and more interesting videos like that. Thank you.